Welcome to the T-cell mediated immunity module. In this talk, we're going to learn about the distribution of T cells in different secondary lymphoid regions. We will first learn about the distribution of different cells in the spleen. These include the lymphocytes as well as other cells that are present in the white pulp. We will then learn about the distribution of lymphocytes in lymph nodes, followed by lymphoid tissues.
We all know that innate immunity is the first immune response that is mounted in response to a pathogen. And innate immune responses can destroy some, but they're not effective against all pathogens. The innate immune responses helps keep the pathogen numbers in check till the adaptive immune system gets activated. We know that innate immunity does not have any immunological memory. And finally, it should be noted that the innate immune response is necessary because it helps in the development of the adaptive immune responses. Now, the adaptive immune responses are mounted later during the course of the infection. However, they are more effective because they can clear most pathogens from the body. Part of the adaptive immune response are the T and B lymphocytes, or T and B cells. Naive T and B cells will be activated in an adaptive immune response, and once they get activated, they become effector cells. We know that B and T cell development occurs in the primary lymphoid organs, and in the case of the B lymphocyte, it occurs in the bone marrow, and in the case of the T lymphocytes, a majority of it occurs in the thymus. Now, when it comes to initiating the adaptive immune responses, these occur in the secondary lymphoid organs. And some examples of secondary lymphoid organs are lymph nodes, spleen, and the mucosa-associated lymphoid tissues, also called as MALT. We've seen this before, but B cells and T cells are organized in specific ways in these secondary lymphoid organs. And this results in B cell zones and T cell zones. So let us look at the organization of B cells and T cells in these secondary lymphoid organs. So let us first look at the organization of the lymphocytes in the spleen. Now, B and T cells are found in the white pulp of the spleen. The B and T cells are able to enter the spleen through these blood vessels that form the marginal sinus, which is what is shown in that black box. The marginal sinus demarcates the white pulp from the red pulp in the spleen. Now, the T cells are able to cluster around the periarteriolar lymphoid sheath, or PALS, and that is what we can see that is shown in these blue stream-like structures that I have outlined with black. So that is where we see a lot of the T cells. Now, where do we see B cells? B cells are basically clustered at the lymphoid follicles, which are these yellowish structures that I have outlined in black. So thus, in the spleen, we are able to see distinct zones where we have the B lymphocytes in the lymphoid follicles, where we have most of the T lymphocytes in the periarteriolar lymphoid sheath. Now, in these B cell and T cell zones, we are able to find other types of cells also. So in the lymphoid follicles, we do find a type of dendritic cells called follicular dendritic cells. Follicular dendritic cells, are also called as FDCs, are different than the bone marrow-derived dendritic cells. The main difference is that follicular dendritic cells are able to capture antigens as immune complexes, and they don't phagocytose the antigen, but however, they simply display the immune complex containing the antigen on their surface. On the other hand, the T cell zones have also a type of dendritic cell called as the interdigitating dendritic cell, which is what we can see in the figure. In the figure, that star-shaped cell is the interdigitating dendritic cell, and all the small circles around it are basically the T lymphocytes. And so it's called as interdigitating because it has these long processes on which the lymphocytes are able to scan through. So thus, in these B cell and T cell zones, in addition to the lymphocytes, we do see other types of cells that aid in the processes of adaptive immunity. Let us now look at another secondary lymphoid organ, which is the lymph nodes. Now, in the case of lymph nodes, the B and T cells are able to enter through a blood vessel called as the high endothelial venule, or HEV. And that is what is shown here in the figure, where we can see the B cell and the T cells entering through the high endothelial venules, and we can also see how they're able to enter into the spleen, where we see a cross section of this high endothelial venule in that black circle that's outlined. Now, once they are in the lymph node, the B cells cluster in these follicles in the cortical region, which is what is shown in black in the figure. On the other hand, the T cells cluster in the paracortical regions, which are shown in that black box and in the blue portion of that lymph node. Now, we do have antigen and antigen presenting cells that have to come to the lymph node to display these different antigens to the B and T cells, and they are able to enter the lymph node through the afferent lymphatic vessels. So the naive B and T cells enter through the blood through the high endothelial venules, whereas the antigens and antigen presenting cells like dendritic cells are entering the lymph node through the lymphatic vessels. The last secondary lymphoid tissue we're going to look at is the MALT, which is the mucosa-associated lymphoid tissue. We can see these lymphoid tissues in our respiratory systems as well as in the gastrointestinal systems, where in the intestines are. Now we're going to look at the Peyer's patches, which is what we see in the intestines, and they also will have B and T cell zones. Now the B and T cells will enter the MALT through the high endothelial venules, which is what is shown in that black circle. Once they enter there, the T cell zones are the portions that lie right beneath the epithelial cells, and that is what is shown in blue in this figure. And we can see that that is right below this box-like pink, which are the intestinal epithelial cells. The B cells are, on the other hand, present in these follicles that are shown in yellow. Now, dendritic cells are also present in the Peyer's patches, and they are able to show or display antigens to the B and T cells.
With this, we come to the end of our talk, where we learned about the distribution of lymphocytes in the spleen, lymph nodes, as well as the mucosa-associated lymphoid tissues.